All right, let's see another example of a condition with damage to the ventral stream. So this will be associative agnosia. It's going to be an old video. It's a patient, a neuropsychology patient being seen by a neuropsychologist, and she's going to test him in various ways to see if he can recognize things visually using visual information just to categorize or recognize objects. So she's testing his ventral stream. She'll put some objects in front of him, and he won't be able to recognize them visually, but you'll see that once he can touch them or smell them, get the information into his brain via a different sensory channel, a different sensory pathway. Like holding an apple, he'll be able to recognize it's an apple. That's through haptics, right? Going through the somatosensory system. Or smelling a crayon or a candle, he'll smell the wax, and with feeling it, he'll be able to recognize what it is. But then she's going to test him on a purely visual task where she just shows a picture of an object. So he can't see it, he can't smell it, he can't taste it, he can't hear it, anything like that. And on this purely visual task, she'll show him something like, I think it's a flute or a clarinet, and you'll see him trying to figure out what the object is. He'll recognize that it's a long object, but you'll see him having struggles trying to identify the object. And then she'll do one where I think she's showing him like a combination lock, like one of those where you spin it around to the right, spin it to the left, that kind of thing. You'll see him get stuck on the old type of telephone, like an old rotary dial type of telephone. So let's watch through this guy as he kind of struggles through these. This is, again, associative agnosia.
Name a few things.
Okay.
What is that?
Apple.
Okay, good. And what is that?
Candle.
Good.
Crayon type stuff, you know.
Okay, which is it?
Crayon. Because that smells better.
Is this a crayon or a
That's
candle?
That's a crayon shaped like a candle. Really?
Well, if it's shaped like a candle, why wouldn't it be a candle?
Because it smells like a crayon.
Oh, I didn't notice you smell. What is that?
It's a pipe.
You can tell by how he's holding it, not how
Okay,
it looks.
good.
Now the visual task, purely visual. Again, she'll show us the picture she's showing him, which is
Okay,
like
I'm
a
going to
flute
show
or a
you this
clarinet.
picture. And I'd like to have you just tell me what you do with that.
Right with it.
I don't want you to tell me. Show me what you do with it.
You're right.
I'm sorry.
It's a pen, you know.
Hold your
Now
hands
watch his
up
hands
here
throughout this
where
whole
we can
thing.
see.
Yeah.
And show me what you do with it.
You're right with it. It's like a ballpoint pen, you know, but it's not a ballpoint pen.
Because he knows it's long. It's a long
Well, take your
object. You can
time
see
and
that.
see if you can show me what you do with
His
this.
hands will figure it out first.
Well, you're right with it, but you know this. I don't know, because I know it doesn't come into focus right. You know, is that a flute or something?
His hands. He was doing
Okay,
the-
that's close. What is that?
I don't know,
How'd you figure that one out?
because that's not the way I put my hands. I know, because I've never been in a flute, and that's the first thing I thought of.
Uh-huh. Did your hands figure out before
Yeah.
before you had it in your mouth?
That's weird. What is that?
That's the clarinet.
Ah, it's close.
That's very close. That's good.
All right, one more. He's going to get stuck on the idea of an old rotary phone, the phone that you spin because it has kind of a circular clock
Another
shape.
thing happens, sometimes you get something stuck in your head,
Yeah, that's true.
okay? So I want you to give up the telephone. It's not a
Okay.
telephone.
Okay. Okay. I'm really at a loss. I can't. It just isn't making any sense
He's
at all
doing something
now.
with his hand.
Is it Bigger than a cat or smaller than a cat?
Than a cat? Well, it's probably smaller than a cat.
About what size is it?
Well, it's about that long.
Mm-hmm. About what size is it? Telephone.
Old phones are big.
Just
About yay by yay.
so this is smaller than a telephone?
Yeah.
Does it have anything to do with communication?
Yeah, because this is where the numbers you gotta hook up with them in order to dial your number or something, you know.
And why do you dial the number?
Because that's evidently what that dial is for. I don't know. Yeah.
Let me give you a hint.
Okay.
I'm going to give you a choice, and you tell me what this is.
Okay, I can tell you then.
Okay. Is this a clock, a lock, or a telephone?
A lock.
Is it a lock?
A clock, a lock, or a telephone? Okay, let's rule out a telephone. A clock or a lock? Clock. That'd be a lock. Must be a lock then.
Are you sure?
No, I'm not. But I don't recognize it as a telephone.
Mm-hmm. Do you recognize it as a lock?
No, honestly, I don't. But it's just it really looks very indescript to me.
But your hands know what they want to do.
I know,
with
and
it.
they want to do something.
That's right.
I can see them. Yeah, I can just picture them, and I know that I have. It's like I'm undoing a combination lock.
Good. Okay, good. We had to work on that one, didn't we?
No shit.
That's associative agnosia. Okay, so those were examples of ventral stream damage, the kinds of things we see in like neuropsychology patients, the functional impairments we see when that lower stream going to the temporal lobe is damaged. We get issues with recognition, categorizing. But I said there's also a second stream, right? A bunch of information comes out of the occipital lobe up and forward to the parietal lobe. So we call this the dorsal stream or the dorsal pathway. You can think of this kind of like, dorsal means like the back or the top. So you can think of it like if you had a dorsal fin on your brain, right? It would be kind of like a shark or a dolphin. It would be on the back here. So the dorsal fin would be kind of up top. That means the dorsal stream is the one that goes up top to the parietal lobe. Sometimes this is called the where pathway, the where stream, or nowadays we call it the where/how pathway, because this is for spatial processing. Like, in other words, taking the visual information and making sense of it spatially. What is to the left of something else in my visual field? What is to the right of something? What are the spatial relationships? What is like movement, right? Movement is going to get processed in this way, visual movement, seeing that something's going from the left to the right, from the top to the bottom. But the reason we call it not just the where pathway, not just the spatial element, but the how pathway, this is also where we get the processing, the use of visual information to do things like reach out and grasp something, to reach out and grab a water bottle so I can bring it to my face, or a spoon so I can navigate it to my mouth to eat. That involves like visually guided movement that is using that occipital information, but sending it to the parietal lobe so we can coordinate it through spatial movement, right? And coordinate with the other spatial information that we get through our somatosensory body information and our motor information up there in M1 of the frontal lobe. So the parietal lobe, really important spatial area of the brain. You can see why it would be processing the spatial elements or spatial functions of vision. Now that means damage to this stream, to the dorsal stream, is going to lead to trouble with spatial stuff, with where or how tasks. So like trouble localizing things in space, not being able to see movement and stuff like that. So I'll give you some examples. Damage to the dorsal stream, it's going to mess with either space or directly acting within space, like reaching out and grabbing things within space, that sort of thing. That's dorsal stream. So if it's damaged, one symptom we might see is something called spatial neglect, where people will only perceive and only sort of act within one side of space. So literally a person might shave half of their face and then be like, okay, I'm done. Like they're looking in the mirror, their eyes work fine, but they just don't seem to notice that one side isn't shaved yet, or only dress half their body. If they look down or looking in a mirror, if they're drawing something, they'll only draw the left half of the object or the left half of the scene. So that's spatial neglect. Sometimes it's called unilateral neglect, because unilateral means one-sided neglect. So it's kind of, we use the same terms for that, but spatial neglect. You're neglecting part of space. You're not able to sort of use vision to process that side of space. There's a different condition, different part of the dorsal stream gets damaged. People can't even copy drawings, so they can't even kind of put together things into the spatial relationships between them. So these are attempts of patients with this type of problem where they're trying to copy the thing up top here. So this is the reference drawing that they're supposed to make a copy of. Some patients get some elements of that, and some are just way the heck off. But basically, they're having trouble keeping those spatial relationships together, right? So they can see the individual lines, but they can't kind of make sense of one line relative to another within space. There's also a condition where people, if a certain part of this pathway gets damaged, like area MT, the sort of motion area in the brain, kind of late in the occipital lobe toward the parietal lobe. If this area gets damaged, people get what's called akinetopsia, where they can't perceive movement or motion. So the root word here, it looks like a crazy word, but just look at the end here. We have opsia. So like optics, like is seeing something, right? Or you go to the optometrist for your eyes. So that's what this last part is like optics. It's like seeing something. So we know it has to do with vision, right? And then we've got that's lack of. So lack of some sort of visual function. And then the middle part here, that's like kinematics or kinesiology major, study of motion, right? So it's lack of motion sight or lack of motion vision. That's where that crazy term comes from, akinetopsia. But it's just like someone who can't perceive the sort of fluid change of the world. It's almost like, how when you're looking at a flip book and someone's flipping through the pictures of the flip book, they're of course just individual images in the flip book. You can look at any page of a flip book and it's just a static image, right? No movement. But if you flip through them quickly enough, our brain puts that together into a sort of motion perception, a stream where everything seems fluidly changing from one part to another. Or when we watch a movie, like the way old film works, when a film is showing on a screen, we see motion, right? We perceive motion, but what it's really doing is just projecting a static image, and then very quickly after that, projecting a slightly different static image. And then a moment later, projecting a slightly different static image. And our brain fuses it together into smooth motion, perceiving movement. And that's what happens when we see our cat running across the room or someone walking towards us for a conversation. Someone with akinetopsia doesn't perceive that. So for example, everything would just kind of jump from one sort of static frame to a very different static frame without a smooth motion between them. So like the person might be trying to fill up a cup and not realize when suddenly it's overflowing because they're not seeing it smoothly fill up a little at a time. They see it empty and then they see it partway full and then suddenly it's overflowing everywhere. And they didn't visually see that happening. The between isn't smooth. So a lot of people with akinetopsia, honestly, you can't really live a normal life. You can't go out and walk through traffic very easily unless you were to put on a blindfold and adjust to blindness and then be able to navigate, because obviously blind people can navigate the world just fine. But if you're trying to do it visually, then you put yourself in danger because you can't see things like a car coming towards you or things like that. Okay, another condition that can happen if part of the dorsal stream of visual input gets damaged is trouble with that sort of, like I said, reaching and grasping. So the how part of the where-how pathway is that being able to just reach out and grab and use objects in space in front of you in a visually guided way. So it's optic ataxia, visual ataxia. So trouble with the movements, but the visually guided movements specifically. So try and reach and grasp something, but they won't be able to grab the right thing. They'll grab right past it. So they'll be reaching out. You can dangle an object in front of the person's face. They reach for it, but they reach off to the side and totally miss it. They can't do that. That would be optic ataxia. And there are other conditions. This is all of these for both the ventral and the dorsal. I'm just giving some examples from neuropsychology. But there's others like simultagnosia, like trouble finding an individual object in a group or an array or in like a whole scene, trouble pulling out individual objects within that scene. Everything kind of just blends together. You have trouble recognizing, oh, that's a set of 5 objects, one in front of another. That's kind of a trouble you can have with dorsal stream damage. Because again, the dorsal stream is taking that visual information and making sense of it within space, or helping us act within space using that visual info. So let's see, this is a simulation. I just found it on YouTube. So it's just someone, it's not a real echinotopsia thing because there's no way to really have you be in that person's eyes or film what they're experiencing. But this is someone's simulation of what it's like based on verbal descriptions. So you'll see kind of normal perception of oncoming traffic.
R31.
Movement. And this is what it might be for someone with echinotopsia. So they can hear the movement, but you don't really see the movement. And this says pouring coffee. They're going to show juice, but you get the idea. So you might be pouring it and you can hear that it's changing, right? And you know that obviously more is going into it, but you don't get the visual feedback in any smooth way. You don't get visual emotion, so you might accidentally overfill. That's an example of what echinotopsia might be like. Again, based on descriptions of people who have that. All right, let's see one more video. This will be for someone with spatial neglect, that one of those other ones I mentioned for dorsal stream damage. So they're kind of unable to visually perceive or at least pay attention to one side of space.
Unlike Graham, Peggy Palmer has normal vision.
She's not bad.
She should be able to copy this star easily.
I'll never get this star, I'm hopeless at this.
But something odd is happening. One whole side of the star is missing. Peggy has a condition called visual neglect. Although her eyesight is fine, half of her visual world no longer seems to matter. 10 years ago, Peggy suffered a stroke in the parietal lobes of her brain.
The parietal lobes are concerned mainly with creating a three-dimensional representation of the spatial layout of the world, allowing a person to walk around, to navigate, to avoid bumping into things. When the right parietal is damaged, the patient is unable to deal with the left side of the world.
This condition has fascinated neurologists for more than a century because it reveals not only how the brain shapes the way we perceive space in the present, it even determines the spatial look of our memories. This became apparent when Peggy was asked to draw a daisy from memory.
Right. A daisy it shall be.
For neuropsychologist Peter Halligan, Peggy's drawings reveal exactly what's gone wrong.
It's like a radar system, whereby the actual radar system on the left-hand side is no longer working well. If someone comes in on my left-hand side now, or I hear a sound, my eyes will immediately move to the left-hand side. That makes me, for evolutionary purposes, very aware of my environment. Because if I wasn't aware of those things, I'd have accidents. I'd get hurt, or I might get eaten by wild animals and whatever. Now, in Peggy's case, she will not attend to those things that we would normally be aware of.
Peggy thinks she's drawn her daisies right, until it's pointed out to her.
You've noticed that, have you?
Oh dear.
So what Peggy's drawn for us is several nice daisies with the left side missing. Same with this one, and this one. And look at this one, this is a very good example.
I've done it on all of them.
Which means that she's not only neglecting events in the world, but when she conjures up a mental image, she's ignoring the left side of that mental image.
Well, I thought I was going all the way round, you see.
And this shows you that this is not simply a sensory problem, but a problem of consciousness.
I don't know. It's because I was so concentrating on that side. It takes everything away, you see. It's this attention, really. It's taken away. There must be two attentions somewhere in your body. That one side's taking the other one away. I can't make it out at all. Very odd.
Peggy's one-sided daisies graphically reveal how damage to the visual centers can warp our consciousness of the world, and how complex the human visual system actually is.
When I was a medical student, I was taught there's an area in the back of the brain called visual cortex, and that's where seeing takes place. But since then we have learned, in fact, there's not just one, there are 30 areas in the brain concerned just with seeing.
For Ramachandran, a walk through this Southern California mall shows exactly what these visual areas have evolved for.
And maybe these different areas are specialized for different aspects of vision. One area for seeing colors, another area for seeing movement, for form and shape, relative distance and depth. Now, despite this staggering complexity of all these different areas, there seems to be a simple overall pattern of organization. In fact, the visual input as it comes in seems to divide into two parallel streams of processing. There is one pathway which we call the how pathway, to which some of these areas belong, and that how pathway seems to be concerned mainly with navigation, with being able to walk around, avoid bumping into obstacles, avoiding uneven terrain, reaching out and grabbing something.
The how pathway leads from the main visual areas to the parietal lobes at the top of the brain, where Peggy suffered her stroke. The other pathway, the what pathway, leads from the main visual areas to the temporal lobes, located just behind our temples.
So it's a ventral stream.
The what pathway is concerned with recognizing the object. What am I looking at? What does it mean for me? Is this an edible object? Is it a flower? Is it a person's face? What is it that I'm looking at, and what does it mean for me? That's what the what pathway is concerned with.
So that was the two streams basically that we see, but again, spatial neglect, that was a dorsal stream issue. Damage to the dorsal stream causes spatial neglect, which makes sense. That's the parietal area, that's the spatial area. Now, and just an important side note to sort of caveat before we go on, those two visual streams, they are actually connected. There are connections between those later parts of the brain, so they do communicate, there is interactions, they send signals all over the place, including some signals going backwards in both of those streams, going back toward V1 and toward the LGN, as we saw before, the part of the thalamus that takes input from our eyes. They send these signals all over the place, including back propagating, where it can help filter, amplify, block, regulate, whatever, the new visual information. So rarely is any function completely localized to activation being in just one single specific place, sort of cut off from other parts of the brain. In reality, brain activity during any task is distributed pretty widely across many areas that connect and communicate. There are just certain areas that seem to have a specific uptick in activity for certain tasks that are selective to those tasks or activate more than to those tasks than to other tasks. And that brings us to the last part of this video, which will be kind of the idea of the brain being modular versus distributed. So modularity of the brain means like that idea that brain regions are specialized for a particular function. We've seen some examples of that already. For example, that fusiform face area seems to be a part of visual recognition processing. So in the temporal lobe, it's in the ventral stream. It's actually pretty late in the ventral stream because we're all the way deep into the temporal lobe at the bottom of it. And there we have an area where those neurons are firing a lot when we put faces in front of the person, like here, like here, like here, and yet not firing much at all, right? This is just sort of a baseline rate of firing or a lower rate of firing when we're showing other things like a place or a plant, outdoor spatial scene, a building, things like that. We get a lot of activity to faces, not so much to non-faces. That seems to be an area that is, in some sense, specialized for faces, so there's modularity there, right? There are other areas that do seem very modular, do seem sort of specific for a task. So another example would be one called the PPA. It's the place area. The first P is actually parahippocampal place area, just meaning it's near the hippocampus. So you may remember, this is a brain from underneath. You may remember the limbic system is in the middle, kind of deep in the brain in the middle. Hippocampus is part of the limbic system. So this is just outside. It's the parahippocampal area. So the parahippocampal place area, you could just remember it as PPA, or place area, but it responds best to spatial layouts. So basically we see activity in this part of the brain when we're showing them pictures like this, pictures that are clearly a sort of spatial layout, a place, right? Like an outdoor place, an indoor place, like the corner of where these walls meet, or even this simulation of a sort of place like built out of Legos. But when we show them other things, like a picture of an object, this area doesn't really light up or doesn't have extra activity beyond its baseline activity. Show them the same kind of thing like Legos, but made into a less of a place and more of just an object, doesn't work. Also, for control conditions, we might take one of the places from the earlier picture and show if maybe it's something about just the colors or the lines or the angles that are in this picture. We can take the picture and just scramble it up and present the pieces of it, but not in that spatial layout, not as a place, but just the pieces individually. And what we find is, nope, this part of the brain, the PPA, doesn't activate when we show that. Likewise, we could take something that does activate for, like this house, but show a scrambled up version, like cut it into little pixels and scramble them up. So we have the same amount of white total in the image, the same amount of dark, the same amount of, like, all the properties of it are basically the same, but scrambled up. And yet we don't find activity here. It has to be a spatial layout, a place. And that causes this one part of the brain to go off. So we have a module that seems specific to helping us recognize places, spatial locations. There's one other area. We won't go through a whole bunch of these. I just want to give a few examples. But there's another area that presents a good example called the EBA. So this is the extrastriate body area. You might just remember it as EBA, or body area. Extrastriate just means it's outside of the striate cortex, which is a way of saying the striped cortex, which is actually our word for V1. V1 is sometimes called the striate cortex because it looks striped. Like if you look at it in a microscope, it looks like stripes because of those orientation columns we talked about in V1. The way all those simple cells were laid out in that perfect little three-dimensional map, that creates striations or stripes through the cortex that you can actually see. So anything that's just outside of the striped area, outside of the striate, is called extrastriate. So it's just visual stuff that's outside of V1, but it's a body area. So outside of V1, body area. And this is for visual recognition of bodies or parts, but doesn't go off for faces. So we can show an arm or someone like this, or a stick figure, or even just a silhouette of a person. All of those will cause this area of the brain, again, there's sort of a left and a right copy. This area of the brain will have extra activity beyond its baseline. But if you show something, even with kind of similar angles, but it's a tool, an object, or a face, or a scrambled up version of the person, or a scrambled up version of the silhouette, it doesn't work. It has to be a body or body part. Basically, we have a specialized area for helping us recognize, oh, this is a body or a biological kind of creature that might come toward us, that might interact with, right? It makes sense why we'd want to be able to recognize that to do things with it. So that is examples of modularity, but it turns out it's a little oversimplified the way we normally describe those as these specialized regions in the brain, because looking at a face or looking at a place like a house, it actually activates many areas in the brain. What we're pointing out when we point out these modules is just that it's strongest in certain areas, or that we have a sort of selective extra amount in certain areas. But really, when it comes to making sense of a face or even making sense of a house, the representation in our brain, just to make sense of what that thing is, to recognize it and use that info, is actually distributed throughout the brain. So stimuli cause neural activity in lots of different areas. Even when we're looking at a face, it's not just the fusiform face area that's active. That's an important area. We can see if it's damaged or doesn't develop properly, we get prosopagnosia, where someone doesn't have that automatic sense of, oh yeah, that's who that is. So clearly, like, the fusiform face area is important for face recognition, but it's not the only area going off. And in fact, most functions that we do day to day require lots of areas activating simultaneously for us to have that function, like face recognition or whatever. So that is distributed across the brain. That's what I mean by distributed representation, that it's not entirely modular or doesn't involve a single module per function. So just going back to the stuff we talked about before, like having a face-specific part of the brain, the fusiform gyrus, right? The FFA, fusiform face area. This is the part of the brain where we see the most activity when we show faces. And again, relative to control conditions, other things like if we show houses or chairs. But when you show houses, you get activity in actually quite a few different areas. In green here are all the areas that are sort of house-specific relative to chairs or faces. The blue areas are what go off for chairs, but not for houses or faces. And the white area is actually areas of the brain that are active when you look at faces or houses or chairs. So maybe just in general, processing visual information. You can see there's quite a bit of overlap, and also it's quite distributed throughout a whole brain. Now, when we say something like the face area of the brain, like the fusiform face area, that it's selective to activating to faces, a lot of people have this idea, either through popular press ideas of neuroscience or even just kind of the standard undergraduate level of understanding neuroscience, often has this misconception that when I say an area of the brain is active for a certain task, the idea you might have is, oh, the brain in that area was off before, like it wasn't firing, and now it's on, like a light switch or like a binary thing. It's either off or it's on. That's not what we're really talking about when we get into this neuroscience stuff. Really what we mean is, all areas of the brain are pretty active when we're just sitting around doing nothing. Like all throughout the day, most of our brain is active, just kind of keeping itself alive. There's a lot of background firing, a lot of baseline firing of neurons all throughout the brain. When I say it goes off, an area of the brain is active when looking at a face, what I really mean is there's an increase in activity there, in neural activity, of maybe a couple percent, 1 or 2%. Or maybe a decrease of 1 or 2%. And those are the changes we're noticing. When you see these fMRI images with blobs of color added in by a computer to tell you where there was extra activity or less activity in a certain condition, that often means a very small change. But that can be enough for us to recognize faces and houses and do all the stuff we do day to day. The brain does a lot of things very efficiently with even very small differences in neural activity. So I just want you to kind of keep that in mind and also recognize, looking at all the areas of the brain that are active for all three of these tasks, so many things we do, basic functions of even like recognizing objects, does involve a lot of neural activity in a lot of different places. So a distributed representation. So I'll give you examples of that, like face processing. I mentioned the fusiform face area. That is part of the brain. So back here, we've got the temporal lobe, right? Back here is the occipital lobe at the very back. The ventral stream is going down from the occipital to the temporal, including along the bottom of it, the inferior part, right? There's the FFA. That's important. That is important for face processing. But look at all these other areas of the brain that are active when we look at a face. So for example, we process the emotional aspects of the face, and that happens with activity going off. When we look at a face, there is often activity in the amygdala, which they've shown with an A here. So we get emotional reactions like recognizing, that's my mom's face, that's a loved one, that's my spouse or something like that, or that's someone I hate, right? You might have a negative emotional reaction to a certain face, but that happens when we see faces. There's activity there, not just in the fusiform face area. Also, the amygdala and nearby areas are important for familiarity, just that sense of like, oh, it's someone I recognize, or oh, it's a stranger. But we're also, when we're looking at a face, automatically your brain starts judging the attractiveness of it. Not just for the purposes of mating, right? It doesn't have to be the type of person you're attracted to, but even just in general, we certainly recognize, or we tend to automatically recognize, like, this is someone I find attractive or someone I don't, or someone I think is sort of objectively attractive or whatever. But your brain is doing that sort of evaluative processing in the frontal lobe when we look at a face. Now that means the information has to come in from your eyes, at the front of your head, comes in from your eyes, goes through the center of your brain, deep inside, because it has to go through the thalamus. Then it goes back to the occipital lobe, specifically V1 back here for initial processing in the occipital cortex, as they've labeled it here. And then it's got to spread to all these areas through the two pathways, right? Through the ventral stream and the dorsal stream. And eventually some of that gets shared with the frontal lobe. So there's a delay of tens of milliseconds or longer, so fractions of a second, before we actually start to be able to evaluate the attractiveness of it. Again, this is sort of automatic, unconscious stuff. We might not even be thinking about the fact that we're doing that. But all these areas get projections from what initially came into the visual cortex, V1. But it goes to all these different areas. We even have a lot of the visual stuff goes to something called the superior temporal sulcus. So superior meaning top instead of inferior, bottom of the temporal lobe, and then sulcus there. So basically at the top of the temporal lobe as it gets close to these other lobes, that's where we use the visual information, but for processing like where someone's looking and how their mouth and face are moving, things like that. All of that goes into face processing. We see activity all across different areas of the brain for something as simple as just, oh, that's my mom's face, or oh, that's my daughter's face, or whatever. That is not just fusiform face area. It doesn't mean that this isn't a specialized area. It is certainly a specialized area, but a lot of other areas go off when we do that function or that task. Same kind of thing. We kind of saw this before when we talked about pain. We get the same distributed activity for other perceptions. Like pain activates multiple places throughout the brain. Like where on the body the pain happened gets processed in S1, primary somatosensory cortex. But also there's the emotional component of pain, how unpleasant it is. That gets processed a lot in the amygdala, the limbic system, areas like that. Learning, memory, same thing. Hippocampus is active. Other stuff like temperature gets processed around there, up there, in the insula, sort of tucked in area that borders kind of tucked underneath the frontal lobe. A lot of areas go off for pain. So any of the functions we experience usually have some distributed activity. Now, the last thing I want to mention is when it comes to these specialized areas of the brain, right? These, whether you want to call them modules or sort of just areas that seem to have a little extra activity during certain functions and not others, a lot of those develop their specialization through a type of plasticity, a learning that happens, like the brain rewiring itself based on our experience, usually meaning based on our experience from the very, very earliest bits of our life, right? When we come out of the womb and start trying to make sense of the jumble of visuals around us. So we call this experience-dependent plasticity, the brain rewiring based on our experience. So there are long and short-term versions of this. So a long-term version of this has been shown in like some classic studies were done on kittens. Again, poor little guys getting experimented on back in the 70s and 80s. It's really sad. But what they did, this is just one type of study they did. But for example, one research study back in the 70s, they took kittens and what they had the pregnant cat in the dark when she was about to give birth. They put her in a dark room so that when the kittens came out, they didn't have any visual experience. So in that first day of life, they weren't able to look around at a bright world and start making sense of the jumble of colors and everything. Instead, to gain experimental control and know that they had not had any other visual experience, the researchers, the experimenters made sure that they were born in total darkness. Then they would take one of the kittens out and go put it in a specialized room, a room that was basically like this big tube here where the walls were all just a vertical stripe, or in a different condition, they might have been just horizontal stripes. Then they turn the lights on. So now this little kitten can wander around in the room and its visual experience, which again is the only visual experience it's ever had in its life so far. Its only visual experience is wandering around this room, going in circles, seeing, for example, this kitten seeing only vertical stripes and nothing else. So they do this for a while, maybe a certain number of days, certain number of hours, whatever you want to do. And then they bring the kitten back into darkness when it's not in the experiment, right? And then they bring it back to this room. So we've controlled its only experiences of vertical lines. That's the only thing it's ever seen. Then they go and do, just like Hubel and Wiesel did that original mapping of V1 neurons, they go back and they measure V1 neurons. They measure simple cells in the primary visual cortex, the ones that go off for lines, right? Lines of certain orientations. They measure those and they look at how many of those V1 simple cells are specialized for, say, straight up and down or mostly up and down lines versus ones that are specialized for side to side horizontal lines. Now, the kittens that were raised in vertical world, right, that had only vertical line experience so far, they had a whole crap ton of simple cells that were specialized for up and down lines and basically no cells at all in their brain. They couldn't find any simple cells in V1 that recognized horizontal lines that had receptive fields. It would only go off when you put a horizontal line in front of them on the screen. They just didn't find neurons that had that specialty in these kittens. The ones that had been raised in a horizontal world, that had only had horizontal experience, they had a bunch of V1 neurons with receptive fields, with RFs that were specialized to horizontal lines, but didn't have any that were really specialized to a vertical line. So that shows that the brain actually develops that wiring, those orientation columns in V1 of all those simple cells laid out in that nice retinotopic map. Like, it develops that over time from our experience. You change the experience, you change that wiring. That's what we mean by experience-dependent plasticity. Now, this was a long-term thing, meaning on the order of quite a while, maybe weeks or whatever. Of course, at the end of this, they did take the kittens, put them back in a totally normal room. They just flipped the lights on in a normal room, let the kittens wander around and have experience with all the normal visual stuff, including side-to-side lines. And sure enough, their brain, they're still young kittens. Their brain is very, very plastic and adaptable. They picked up like normal cells. Everything got back to normal and they developed normally. They had no difference from control kittens later on. So there seemed to be no lasting impact or damage from this. But I just wanted to point that out because obviously it's kind of an ethically sketchy thing, especially if you like cats. But in this case, it didn't cause any lasting damage to them. Okay, there are also short-term versions of experience-dependent plasticity that we've shown in, for example, experiments with human subjects where one famous example of them had these little kind of clay figures, these little sculptures that they called greebles, just a random word for these little, I don't know, alien or bird-like heads, whatever you want to think of these as. But they took these little sculptures and they had people look at pictures like this versus looking at pictures of real human faces while they were in a brain scanner. So like an fMRI machine, something like that. And before any other thing, so just putting faces in front of people, that's our red line here, faces in front of people, you get a lot of activity in the part of the brain we would expect. The fusiform face area lights up a whole bunch. Not so much when you present the greebles. So when they see these pictures, right, the blue line here is the greebles, these little guys. No real activity, or very little anyway, very little activity in the fusiform face area. Then they did the intervention. They had people practice attaching a name to these. They give them a name for each of these different shapes, right? They all look kind of similar, but they're different enough you can tell them apart. They had people practice attaching a name to different ones, so they'd have to recognize to tell this one apart from this one apart from this one, and so on. After that training, they went back, put these people in the brain scanner, and once again would put faces or greebles in front of them. And sure enough, we get, there's statistically no difference between these two bars, but we get activity for both faces and greebles in this case. So there's a change in what the fusiform face area is specialized to as we have more experience with greebles. This has actually been shown, a similar thing with people who work with sheep, for example, on a farm. Someone who works a whole bunch with sheep their whole lives can tell individual sheep faces apart in a way that someone who hasn't worked on a farm can't tell different sheep faces apart as easily. That comes from experience, right? And that is plasticity. Our brain can actually pick up on that, and it uses the fusiform face area to do that. Okay, taking that, we could say, well, what if we measure, let's say, activity in the fusiform face area, the FFA, when people are looking at, I don't know, bird pictures or car pictures or radiology scans, like looking for tumors, or looking at a chessboard, would you expect activity in the fusiform face area for those things? Probably not, because they're not like faces, right? Those greebles, they looked a little like faces. So it makes sense how we could train the FFA to kind of start picking up on those and have an activity for those. But certainly not for birds or cars or whatever, right? Except, turns out, if you measure a bird expert, someone who's like a bird watcher or an ornithologist, they have increased FFA activity when looking at birds relative to control subjects who haven't worked with birds their whole lives. Car experts, so someone who's a mechanic, has increased FFA activity to, that should say cars. So to cars, someone who's worked with them their whole lives, they actually have FFA activity going on when they're looking at cars, which certainly aren't faces, but just sort of visual recognition expertise, right? Radiologists who spend their whole lives looking at these scans, making slight visual distinctions between this scan or this scan to pick out a tumor, something like that, they have the fusiform face active area going off when they look at scans. chess masters, so those who are like a grandmaster at chess, they've been doing it for most of their life, they also have FFA activity when they're playing chess. So really the fusiform face area, we call it the fusiform face area because that's a pretty universal use of that area. It makes sense. We all come out and immediately start looking at faces. First thing first, usually the parent is holding the child up to the face. That's like what they see the most. So we have an area of the brain that's just ready to pick up on patterns of, things we see very commonly. And some people have started to say, instead of calling this the fusiform face area, we could really start thinking of it as like a visual expertise area. And that's a topic that's sort of still debated today, but certainly we found a lot of evidence that suggests this area isn't only for faces. It just tends to be specialized in faces because that's something we pretty much all have lots of visual expertise experience with. But it can go off for other things if you spend a lot of time distinguishing them in the same way that we make these subtle distinctions between faces. That would be experience-dependent plasticity. It's a modular area of the brain, right? A face module, so to speak, but turns out there's some plasticity or some flexibility about what it really is specialized for, and that function can be changed based on our experience. Just to kind of put it in more timely, there's a study that came out in 2019, not too long ago, showing that people who played Pokemon in their childhood, like played a whole bunch of Pokemon, they have sort of reorganized the fusiform face area and actually other areas of the visual cortex that shows this experience-dependent plasticity. So their brain had wired based on practicing distinguishing these different shapes of Pokemons. That actually has this lasting effect in terms of how their brain wired itself for how they recognize those things later, what parts of the brain do that. So, okay, we will stop this video for now on these dual streams and different parts of the brain that are specialized for these high-level functions, things like the fusiform face area, extrastriate body area, parahippocampal place area. We're going to move on in the next video to sort of higher level object perception, object recognition, and perceiving objects, and some of the challenges that go along with that for our brain.